Good morning students. So last class we discussed about chapter 8 microbes and the disease caused by the microorganism. So in this chapter, this is chapter 10, okay. So it is an interrelationship between chapter 8 and chapter 10, that is the microbes in human welfare, okay. So here we are going to see how the microorganisms are playing an important role in the human beings in our day to day life, how it is giving importance to us, okay. So as we know earlier, we are having in the diversity, we are having macroorganisms as well as the microorganisms. So these microorganisms are omnipresent. Omnipresent means it is present everywhere. So even if you take this room, in this room also the microorganisms will be present, okay. So if you go outside, the outside atmospheric air also microorganisms will be found, okay. So it is omnipresent, it is present everywhere, even in the sea, even in the volcano, some microorganisms can able to tolerate and grow, okay. So that organisms are said to be thermophiles because they can able to obtain, they can able to sustain even when there is a high temperature, okay. So even in sea, if you take sea water, it will be very salty, right. So that organisms are said to be halotolerant. Halo means salt, okay. So the salt tolerant microorganisms can also live even in the surface of the sea water or in the deep sea also, okay. In the submerged region also microorganisms will survive, okay. So we may think that how the microorganisms are surviving. For example, if you take the human beings or plants or animals, we are getting sources, okay. So we are getting oxygen from the environment, plants are getting sunlight from the sun, okay. So how the microorganisms present in the deep sea can able to survive? So because of the absence of oxygen also the microorganisms can able to survive. How it can able to survive? That process is said to be the fermentation process. So we are saying two types of anaerobes based upon the utilization of oxygen, okay. So one is said to be aerobe and another one is said to be anaerobes. Aerobes and anaerobes. So aerobes means the microorganisms which can able to live in the presence of oxygen, that is said to be aerobic organism. Anaerobes means the microorganisms can able to live even in the absence of CO2, that is said to be the anaerobes, okay. So what is happening? So in the presence of oxygen, how the humans can able to respire? We are respiring the oxygen and we are converting the glucose molecules in order to produce the ATP, okay. The production of ATP plays an important role for any organisms to survive. So that is happening in case of aerobes. Even in case of anaerobes also the same ATP molecules has to be produced, okay. The same anaerobes is also taking glucose molecule, that is we are saying it as C6H12O6 is said to be the glucose. So that glucose, even in the absence of oxygen also, some bacteria can able to convert this glucose molecules into C2H5OH, it is said to be ethanol and CO2, okay. So 1 molar glucose is converted into 2 molecules of ethanol as well as 2 molecules of carbon dioxide. So this is the process which we are saying it as a fermentation. That is, fermentation means it is a basic metabolic process which converts the sugar molecules into alcohol as well as some gases, okay. So this is the common representation of your fermentation, okay. So this can be vary if you are using some other bacteria, each and every bacteria can vary the presence, okay. If you are going for some other bacteria like Clostridium, okay. So if the Clostridium organism is there, means it will utilize the glucose and it will produce butyric acid instead of CO2, okay. Whatever the reaction may be, the end product will be one acid and a gas, okay. This is said to be the fermentation. So in simple we can say fermentation is a metabolic process in which the end product will be our acid as well as alcohol as well as the ATP molecules, okay. The ATP molecules will get released finally and our bacteria can able to survive. That is said to be the fermentation process. The fermentation process also can be classified into two types. One is said to be aerobic fermentation and another one is said to be anaerobic fermentation. We know that aerobic fermentation means in the presence of oxygen this will convert, okay. Normal our glycolysis process, that is said to be the aerobic fermentation. So anaerobic fermentation means in the absence of oxygen, so So our bacteria can able to convert these glucose molecules into simpler alcohol as well as some other gas molecules will be released. Okay. So this is the basic term for fermentation. So the container in which the fermentation process will be takes place is said to be the fermenter. Okay. So in industries we are applying this fermentation process and we are producing antibiotics, we are producing acids, we are producing enzymes in large quantities. Okay. So for large quantities of production we are in need of a large equipment. So that equipment is said to be the fermenter. Fermenter is an equipment in which the fermentation process will carry out. Okay. So now we are going to see the application of this fermentation in our day to day life. Okay. So first one is microbes in our household. So we can take an example, curd. So what is a curd? We know that, okay, so curd means how it is forming. So with the help of milk we are producing the curd. So what they will do in our home for preparing the curd, they will take the previous day curd. Okay. So that curd is said to be the inoculum. We have to know the word inoculum. Inoculum means a simple or a group of microorganisms which can be added to the reaction in order to start the reaction. That is said to be the inoculum. Inoculum means a small quantity of a substance, okay, which contains the microorganisms. If you take the previous day curd, 1 spoon of curd, they will add it in the milk, okay, and they will mix it and they will keep it for 8 to 9 hours. So after 8 hours we can able to see the curd formation. Our complete milk has converted into the curd. Okay. So that is also one process of fermentation. The agent which we are adding, that is said to be the inoculum, okay, which is nothing but a old day curd. Okay. So see here what is happening in the formation of curd means, so milk. So in our milk we are having some protein molecules. The important protein molecules are said to be the casein. Okay. So milk is also made up of some starch materials, that is we are saying it is lactose, some sugar molecules, we are saying that one as lactose. In milk we are having a large amount of lactose. Okay. So what is happening means we are adding the inoculum, right, we are adding the old day curd. Okay. So that contain a bacteria lactobacillus. So lactobacillus is the bacteria which is present in the previous day curd. Okay. So this lactobacillus will convert lactose into lactic acid. As I have told earlier, glucose is converted into ethanol, okay, or some other acids. The same principle here. So this lactose, our lactobacillus is converting this lactose into lactic acid. Okay. So in our milk, now the milk is a basic condition, it is now converted into an acidic condition. So what will happen if acid is present means, so it will go and act on the enzymes. Casein is a protein, proteins are nothing but an enzyme. Okay. So it will act on the casein molecules. This lactic acid will go and act on the casein molecule, and this casein will get curdles. Curdles means it will form a coagulant. Okay. So finally our end product curd we can able to get. This is the basic principles in the formation of curd. So we are having lactose in the milk. So if you are adding the inoculum, that is the old day curd, means what will happen, the lactose is converting into lactic acid and our milk is turning into sour taste. Okay. So then that sour milk acted on this casein present in the milk and it will coagulate halfly and forms a curdles. We can able to get the curd. Okay. So this is the basic reaction in our day to day life we are doing. Okay. So we can also take an example of dosa or idli batter. So what we are doing, we are grinding the black gram as well as the rice. Okay. And we are mixing it together with the help of salt. Clear. So with the help of salt we are mixing and we are keeping it for 8 hours in the room temperature. So one thing is you have to keep a particular temperature. Temperature plays an important role in fermentation process. Okay. So here we are adding the rice plus black gram. Okay. So we are mixing it with the salt and we are keeping it for 8 to 10 hours. Okay. So the next day morning what we are seeing in the dosa batter, we can able to see some foamy layers. Okay. So it will bulge. Okay, suppose I am keeping the dosa batter at this level, means the next day morning if you see it will rise to this level because of the presence of CO2. So here what has happened means fermentation has happened. So we didn't add any inoculum. So how the fermentation happened means the organisms present in the environment, okay. So or some organisms may be present in my hand also. So I am mixing the batter with the help of hand, okay. So that is why in olden days they used hand for mixing these batters. So if I mix it with the hand, so my body temperature will be inoculated in this batter. So with the help of that temperature, the batter can be able to ferment and it can be able to produce the carbon dioxide, okay. So this is the basic principle for our idli dosa batter, and we can also made adirasam, okay. So that is also happening only in this principle. The organisms present in the atmosphere will come and attach to that batter and it will convert into a simpler molecules with the release of CO2. That is why we are getting the puffness in the batter, clear. The same principle they are following in the brewing industry, but with the help of yeast, okay. So here baker's yeast plays an important role, that is the Saccharomyces cerevisiae. We are saying Saccharomyces cerevisiae as a baker's yeast or brewer's yeast, okay. So they are applying it in the industries, okay. So the industries means mainly it will used in the bakery industries, okay. So they are mixing the dough with the help of yeast which is readily available in our market, okay. So by giving a particular temperature or by the presence of sugar molecules, so in that batter our yeast will convert into an acid or some other gas will be produced, okay. So that only in bun and cake we are getting the softness, okay. So because of that chemical reaction, because of that fermentation reaction only, the softness will be appears in the bun or in the cake molecules, okay. The same principle they are following for cheese as well as for butter, okay. So here the cheese they are classifying it into two types. One is said to be the Swiss cheese and another one is said to be Roquefort cheese. First we will see Swiss cheese, okay. So here in the Swiss cheese there will be a large holes present in the Swiss cheese, okay. So why the holes happening means, as I told earlier, because of the presence of CO2 molecule, because of the release of CO2 molecule, okay. So here the Propionibacterium shermani, okay, this is the bacteria, they are using it for the production of cheese molecule, okay. Roquefort cheese, so this is also similar to the Swiss cheese, okay. So here after inoculating the bacteria, they are inoculating the fungus particle, that is said to be Penicillium camemberti, okay. So these fungus particles will act on the Swiss cheese and it is converting the Swiss cheese into a blue color, okay. So the blue color formation will be due to the presence of the addition of the Penicillium camemberti, okay. So the Swiss cheese is converted into Roquefort cheese. So what is the difference, main difference between Swiss and Roquefort cheese means, here bacteria is involving, so here including the bacteria we are involving the fungus particle also, okay. So this fungus molecules will give some flavor and color, extra flavor and color to our Roquefort cheese, okay. So this coming to the industrial products, so these are all the household thing, okay. So we can able to prepare curd, dosa batter and some bakery products, bun, okay. So these are all important household things. Now we are going to see the industrial products. So coming to the industrial products, the main thing is beverages and antibiotics. So beverages means which includes rum, wine, brandy, whiskey, vodka, so everything will come under beverages. So how they are preparing the wine, rum, everything means with the help of our baker's yeast, okay. So the Saccharomyces cerevisiae, okay, this is acting as a Saccharomyces cerevisiae, okay. This is said to be the top yeast. We can give another name also, we can give it as top yeast, brewer's yeast, baker's yeast, okay. So this Saccharomyces cerevisiae plays an important role in our beverage industry, okay. So for the production of wine, for the production of alcohol, so for the production of rum, everything we are using only this yeast. So what this yeast will do means, for example, if you take wine as an example, we can produce even wine in the foam itself, okay. So we can take the grape and we can wash it, make it as a juice and fill it in the container, okay, glass container which is having a lid, okay. So we are adding the grape juice and we are inoculating the yeast. So yeast, I have told already, it is available in the market in small round particles, okay. So we can add the yeast along with little sugar. So why we are adding sugar when adding the yeast means in order to initiate the reaction very fast, okay, because sugar is made up of glucose or sucrose molecules. So the yeast will uptake the glucose molecules and it will convert into an acid, okay, plus CO2. Some CO2 will be eliminated. So if you are keeping that setup for 1 month means we can able to get a clarified wine. The top bottom, in the bottle, the top bottom is said to be the wine, okay. We can filter it and we can drink. So it is having a small amount of alcohol, only 8% of alcohol if you prepare one with the help of a grape, okay. So these are all the basic brewing processes. So if we are doing the brewing process in the industries means only in the fermenter we can able to do, okay. So the fermenter which is having a closed appearance, the agitator will be present, okay. So agitator means which is used for aeration, okay. So it is used for aeration as well as proper mixing. So if you are going for beverage industry you can see the fermenter, right. So the next one is antibiotics producer. These are all the industrial applications. So coming to the antibiotics, what is mean by antibiotics? We know that antibiotic means a chemical substance or a substance produced by the microorganisms or plants which can able to kill the other organism. That is said to be the antibiotics, a chemical substance which can able to kill the pathogenic organisms is said to be the antibiotics, okay. So this antibiotics was first discovered by Alexander Fleming, okay. Later it was studied by Ernest Chain and Howard, okay. These three scientists got Nobel Prize in the year 1945 for the discovery of antibiotics, okay. So the first antibiotics which was discovered was penicillin. It is just an accidental discovery. So I will tell how the discovery happened, okay. So once Alexander Fleming, he was working in the bacteria Staphylococcus, okay. Staphylococcus means it is a bacteria which is cockeye shape, round shape. So that is why we are saying it is Staphylococcus, okay. So he grew this bacteria in a Petri plate. Petri plate means it is a plate where the artificial nutrients will be present, okay. So he grew this bacteria in the Petri plate, okay. So he left the bacteria without unwashing for a week, okay. So he left it as such, okay. So what had happened means he found that in a particular region some group of colonies was present, okay. So he found in a particular region some group of colonies were found. So in that region there is no presence of Staphylococcus aureus, okay. So here if you take a Staphylococcus, if you are growing it in the medium means what will happen normally, if we are growing the bacteria in the medium, it will form a colonies and it will grow throughout the plate, okay. We can see it visibly, right. So he found that in a particular region there is no growth of Staphylococcus aureus. So instead he found one mold was grown, that is called as penicillium. Later he named it as penicillium, okay. So what happened here means there is no growth of Staphylococcus aureus. So what happened here means this penicillium, it is producing some chemical substance, okay. So that chemical substance we have named it as penicillin. So this chemical substance will inhibit the growth of Staphylococcus aureus, okay. So what is happening? Penicillium grown in the medium. So if we left idli for 1 week, what will happen? We can see the black color mold around the idli, okay. So that is nothing but a fungus. The same happened here. He left the plate as such, okay. So after 1 week he found that some mold was present, but in the nearby region of the mold there is no Staphylococcus aureus. In the remaining region there is a presence of Staphylococcus, okay. He actually, the species name here is Staphylococcus aureus. The bacterial species name is aureus. So he found there is no aureus growth, that is no Staphylococcus growth in the region of penicillin growth. So what he found means the chemical substance released by the penicillium, that is the penicillin, will act on the Staphylococcus aureus. So it can able to inhibit the growth of Staphylococcus aureus, okay. So this discovery paved the way for our antibiotic penicillin, okay. So this is the first antibiotics which was discovered by our scientist Alexander Fleming, and he got the Nobel Prize in 1945. Okay. And one more point, the fermentation I have told na, aerobic fermentation as well as anaerobic fermentation. So those was first discovered by the scientist Louis Pasteur. Okay. So in Kunnur we are having a Pasteur Research Institute. So what they are doing means they are producing the penicillin antibiotics, not only penicillin antibiotics, they are producing so many antibiotics. If you take the polio vaccine, so everything they are preparing in a large quantities in Ooty, Kunnur. Okay. After the name of Louis Pasteur only, they have told that they have named it as Louis Pasteur Institute. Okay. So now coming to the applications. So other applications we are having is enzyme production as well as the bioactive molecules. Apart from the industrial and household products, our microorganisms also plays an important role in industries. Okay, so it is giving important acid molecules. So these are all the microorganisms you have to remember in mind. Okay, so write the names twice or thrice, then only you can able to memorize the spelling of the genus as well as the species. You should not go wrong in the spelling. Okay, so for example, if you take Aspergillus niger, okay, so it is a fungus, so it is helpful for the production of citric acid. So we know the application of citric acid, so it is acting as an important agent in the food preservative agency. Okay, so citric acid as well as acetic acid, they are using as a food preservatives. So in case of acetic acid, which organism is producing means Acetobacter aceti. Okay, so this is a bacteria which can able to produce the acetic acid, and also these organisms, these acids plays an important role in emulsions, paint preparations. Okay, so butyric acid and all playing an important role in paint preparation. So this was carried out by the organism Clostridium botulinum. Okay, botulicum. Okay, so this is the organism which can able to convert the starch molecules. So here we are using what the raw material we are using means we are using the sugarcane bagasse, that is the leftover sugarcane. Okay, after taking the sugarcane juice we are putting the sugarcane waste as such. Okay, so they are taking the sugarcane waste, that is said to be the bagasse. So from that bagasse we are producing these acids. Okay, they are dumping they will dump the bagasse in a fermenter in a large quantities and they will add the inoculum. Inoculum means this particular microorganism, they will add it, they will give a particular temperature. Okay, so by giving the particular temperature and particular organisms, we can able to get the specific acids, citric acid, acetic acid as well as the butyric acids. And also we know very well that lactobacillus will produce lactic acid. Mainly the species Lactobacillus lacti is responsible for producing the lactic acids, and also enzymes we can produce it. We are using a daily life, we are using a detergent, right? So in our detergent we are having an enzyme lipase. So some microorganisms can able to produce the enzyme lipase. So we are isolating the lipase enzyme from the organism in a large quantities and we are using it for our laundry purpose, that is it will remove the stains in our dress. Okay. The next one is protease enzyme. Protease as well as pectinase, these are all the enzymes which are used for clarifying the juices. Okay. If you see the juices which is available in the market in a bottled form, so it will be very pure in nature. Okay. So how this clarification happens means by adding the enzyme proteases as well as pectinases. Okay. These are all the industrial applications, and some medical applications are also there apart from the antibiotics. That is, we know that cyclosporin, it is a medicine available from the fungus Trichoderma polysporum. Okay, Trichoderma is a fungus which is also used in medical as well as we are using it as a biopesticide also. I will tell it little later. So Trichoderma polysporum. So you have to remember the name correctly, which can able to produce a compound cyclosporin A. So this cyclosporin A is acting as a immunosuppressor. Immunosuppressor. In chapter 8 we have discussed about transplantation, right? So if kidney is transplanting from one patient to other patient, that kidney can be identified as a foreign antigen, right? So in order to suppress their immune system for the transplanted patients, they will give the drug cyclosporin. Okay, so this drug will prevent the patient from graft rejection. Graft means the transplanted kidney or the transplanted heart. Okay, whatever the organ they are transplanting, that is said to be the graft. Okay, so for that purpose they are using the cyclosporin A. The next one is Monocercus purpurians. So here this is a very important compound, very important organism. Okay, this is said to be the yeast. This yeast is producing statins. Statin is a chemical compound which are producing as a byproduct of this yeast molecule while producing the alcohol. We know that yeast undergoes fermentation and produces alcohol, right. So in between one chemical product is producing, that is said to be the statins. So this statin is acting as a reduction of blood cholesterol level. So what it is doing means, this statin will go and inhibit the enzyme. Okay, the statin is a chemical compound obtained from the yeast monascus. So this will convert, this will suppress the enzyme, okay, which is responsible for the production of cholesterol. So if a patient is having a high cholesterol, means how the cholesterol is producing with the help of an enzyme, okay. So that particular enzyme was suppressed, okay, with the help of the chemical statin. So statin plays an important role in medical field. And also another one compound, streptokinase, okay. So this is very important for curing the myocardial infarction. So from the bacteria Streptococcus, we are isolating an enzyme streptokinase, okay. So this enzyme is act as a clot buster. We are saying this as a clot buster. So why we are saying this means, this will prevent the clot. So if a patient is having a clot in the heart vessel means, so if we are giving the streptokinase enzyme means, so it will remove the clot, okay. So this is an important application in the myocardial infected patients, okay. So these microorganisms plays a very important role in our day to day life, okay, and also industrial applications and also some medical applications, okay. So kindly go through this name of genus and species carefully and write it in your notebook. So in your book they have given like a cluster, okay. So I have divided it for your understanding. So based upon this, you write it in your rough notebook and you study this, okay. Each and every organism is very important for your examination, okay. So next we will see it in the next class. Thank you.